Now that we have covered cell replication and the ideas of chromosomes and genes, we will take it one step further in Chapter 8, where we will discuss how genes are passed down from parent to offspring. To begin the chapter, understand that heredity is when genetic material is passed from parent to offspring, while genetics is the science of inheritance. Notice the diagram. The father and mother have different traits, and they both pass traits to kids. We will look at how traits are inherited and why sometimes traits do not show themselves. To understand heredity, it is very important to know these terms. Genes are sections on a chromosome and will be in the same location or locus of the two homologous chromosomes inherited from both parents. Alleles are variations in traits. Homologous chromosomes can have the same allele or they can have different alleles. In the top example, notice the purple and green bands are the locus or location of flower color. The left chromosome has the allele for white flowers, and the white chromosome has the allele for purple flowers. The two chromosomes are homologous, meaning one came from each parent, and they match each other in length, location of centromere, the bottleneck area, and the loci of the genes. In the bottom example, the two homologous chromosomes show two genes. These have the same allele for plant height and a different allele for the texture of the pea. This allele arrangement depends entirely on what genetics the parents pass on to the offspring. What is the actual difference in two alleles? Notice the dominant smooth pea versus the recessive wrinkled pea in the left image. The left half of the other image shows the DNA sequence of 3,550 nucleotides for the dominant allele of smooth pea shape. For the recessive wrinkled version of the same gene, the red portion of the DNA sequence is an insertion of over 800 nucleotides. This insertion in the middle of the gene sequence still allows the plant to produce a pea shape, but changes the structure of the pea, preventing it from having a firm shape. So alleles are variations of the same gene that may or may not be expressed depending on inheritance. Long before scientists knew anything about chromosomes, genes, or alleles, a monk named Gregor Mendel was curious about patterns of inheritance. He studied insects and plants, doing most of his work specifically with pea plants. Mendel is considered the father of genetics, although his work was not recognized during his lifetime. During the time when he studied pea plants, the common inheritance belief was that offspring were a mixture of the parents' traits, an idea known as continuous variation, where only slight variations occur within populations. However, Mendel's work with pea plant traits showed that offspring are not always a blend of the parental traits, but that variations in the traits of offspring can occur, which is known as discontinuous variation, such as when two purple flowers produce offspring with white flowers. Mendel's work was rediscovered years later and is still the foundation of modern genetic theories. Mendel chose to use the pea plant as it self-pollinated and therefore created true breeding plants where offspring were identical to the parent plants. However, Mendel created hybrid plants by manually transferring pollen, which is the sperm, from one variety of plant to the stigma or the egg of another plant. The hybrid that had resulted had a combination of genetics from parents who are not similar to each other in at least one trait. Hybrids can result in a variety of outcomes. When one tall plant is crossed with a short plant, 3 variations might result: a tall plant, a short plant, or possibly even a medium-sized plant depending on the genetics of the parents. We will discuss this last variation later in the chapter. When discussing generations of hybrids, the P generation is the original parent generation. The first crossbred offspring are referred to as the F1 generation, and the cross of two F1 plants produce in F2 generation. The F stands for filial, which means sons and daughters. When Mendel chose plants to crossbreed, he picked F1 hybrids where one trait from a parent was masked by a trait of the other parent. He would then cross two F1 offspring to produce F2 offspring. When he did this, some of the masked traits of the F1 generation were expressed again in the F2 generation. This process allowed him to better illustrate that not all parental traits were lost after the first breeding. For example, look at the diagram. When violet-flowered parent plants were crossed with white-flowered parent plants, the offspring were 100% violet, appearing as if the white gene had disappeared. He would then allow two F1 hybrids to self-pollinate, producing the F2 offspring. In the F2 generation, two variations resulted again, violet and white, in a 3-to-1 ratio, 75% purple to 25% white. This became the foundation of Mendel's work. Mendel's pea plants had specific characteristics he could measure from generation to generation. These physical characteristics are known as traits. He studied traits such as plant height, texture of seed, color of seed, pea pod size, location of flowers, color of flowers, etc. As Mendel studied the traits of pea plants, he determined it appeared that one trait, such as white flowers, completely disappeared in the F1 hybrids. So he flipped the experiment in what is known as a reciprocal cross. He took pollen from the white flower and manually put it in the violet flower, opposite of his first experiments, but got the same exact results. Notice in the image, in the first set of crosses, the pollen, which is the male portion of the plant, is placed on a green stigma or egg in another plant, resulting in only yellow offspring. In the second cross, the color of the pollen and stigma are flipped, but the results are the same in the offspring. This is an example of a reciprocal cross. Dominant alleles are traits that will be expressed if inherited. Recessive traits are only expressed when both recessive copies of the allele are inherited. A dominant allele will mask the expression of a recessive allele. Homozygous refers to two traits that are the same or identical, while heterozygous means a combination of dominant and recessive. When using the term homozygous, the term dominant or recessive is also used to clarify which matching sets of alleles were inherited. For example, if a person inherits two dominant alleles for eye color from both parents and therefore has brown eyes, they are said to be homozygous dominant, which would be written capital B, capital B. If a person inherits both recessive eye color genes and has blue eyes, they are homozygous recessive, which is written lowercase b, lowercase b. However, if a person inherits one dominant allele from one parent and one recessive allele from the other parent, they are heterozygous for eye color, but will express brown eyes because the brown is dominant and masks the blue. The combination is written capital B, lowercase b. This follows Mendel's law of dominance, stating that with heterozygous traits, the dominant will mask the recessive. Traits can be discussed in either a phenotypic or a genotypic manner. The phenotype of a trait is what is physically expressed. In eyes, this would be brown, green, blue, etc. The genotype would be the molecular DNA on the chromosome and is written as a set of letters as discussed in the previous slide. For example, the phenotypes, what is physically visible, of seed color in two plants are yellow and green. If yellow is dominant and therefore masks the green, it is written with capital letters. The green seed is recessive, which can be masked, and is written with lowercase letters. The same letter is used for both alleles to avoid confusion, such as in this example, the letter Y is used, so dominant yellow is capital Y and recessive green is lowercase y. Notice in the F2 generation under genotype, both the homozygous dominant, which is 2 capital Y's, and the heterozygous, which is 1 capital 1 lowercase y, both offspring express as yellow because the dominant allele is present, so it will mask the recessive green allele. Here is the example of eye color genetics once again to illustrate dominance and zygosity. When a person has the physical trait or phenotype of brown eyes, they could have one of two genotypes because brown is dominant over blue. Notice in the diagram the homozygous dominant, or 2 capital B's, and the heterozygous, or capital B lowercase b, genotypes are both expressed as brown, while it takes 2 recessive blue alleles, or homozygous recessive, to produce the phenotype of blue eyes. Please understand, eye color inheritance is much more complicated than just this image just depicts. In reality, eye color is impacted by at least 8 genes leading to a range of colors. Two parents that are true breeding, homozygous, either dominant or homozygous recessive, but differ by only one trait, can be crossed in a monohybrid cross. These crosses produce offspring known as monohybrids. In the image on the left, the two parent guinea pigs are both homozygous. One is dominant while one is recessive. Notice the genotypes of capital B and lowercase b above the parents. When they are crossed, they each pass down one allele thanks to meiosis cell division, and all offspring receive one dominant and one recessive allele, resulting in heterozygotic offspring. However, notice that the heterozygote is black like the dominant genotype of the female parent. In the right image, when two heterozygous parents are crossed, the possible outcomes are homozygous dominant, heterozygous, or homozygous recessive. Notice, however, that the homozygous dominant and heterozygous offspring all express phenotypes of black fur, while only the homozygous recessive offspring express the brown fur. The square with 4 outcomes on the right is called a Punnett square and is used in genetics to determine probabilities of genetic outcomes. Watch the linked video for more on monohybrid crosses. Punnett squares help visualize the outcome of allele combinations in offspring. One parent's alleles are placed across the top while the other parent's alleles are placed along the left side. It does not matter on which side a parent is placed as the results are the same. In this example of red versus white eyes, one parent is homozygous dominant with red eyes and the other parent is homozygous recessive with white eyes. The squares are filled by transferring each allele across and down. When the alleles have been filled in for the possible combinations, probabilities can be determined. In this case, there is a 25% chance of homozygous dominant offspring, 50% chance for heterozygous offspring, and 25% chance for homozygous recessive offspring. And when we're talking about phenotypes, what you could actually physically see, there's a 75% chance of red eyes, but only a 25% chance for white eyes. Watch the linked video for an example of a Punnett square being completed. Although Mendel did not know about meiosis, he proposed an idea known as the law of segregation, where genes split equally into different gametes, resulting in an equal chance of inheriting either allele. When an organism's phenotype expresses the dominant form of a trait, it is not always possible to know the genotype. Is it homozygous dominant or heterozygous, where that dominant allele is masking the recessive allele. A test cross will determine the genotype of an unknown subject. Notice in the image on the top, the yellow seed is the unknown. Is it homozygous dominant or heterozygous? The green is known as it can only be green if it carries both recessive alleles. If the unknown subject is crossed with a green recessive plant, it will reveal the genotype of the unknown. In the top example, if the unknown is crossed with a homozygous recessive and produces only heterozygous offspring, it has to have been homozygous dominant, 2 capital Y's, in order for all 4 possibilities to have received a dominant allele. However, if the outcome is like the bottom box, where half of the chances are for heterozygous and half are for recessive, then the unknown must have had a recessive allele to pass along, meaning the unknown is heterozygous. Watch the linked video for an animation of this idea. One chromosome can carry hundreds or even thousands of different genes, but Mendel believed that the inheritance of one trait did not necessarily influence the inheritance of other traits. This idea is known as the law of independent assortment and explains how traits could be inherited independently of each other. As a result of meiosis, as well as crossing over genetic material during prophase, random assortment is possible. This idea can be tested using a dihybrid cross where 2 traits are tested instead of 1. A dihybrid cross uses 2 sets of alleles from 2 chromosomes to demonstrate independent inheritance. To determine the allele combinations of a dihybrid cross, the FOIL method can be used. Matching the first letter of each gene, in this case capital A and capital B, then the 2 outer alleles, capital A, lowercase b, then the 2 inner ones, lowercase a, capital B, and finally the last of both genes, lowercase a and lowercase b, will show the possible outcomes of what alleles could end up in the gametes of this dihybrid genotype. When these 4 possible combinations for one parent are put into a Punnett square and crossed against the 4 combinations from the other parent, it is possible to determine the probability of the outcome of dihybrid crosses. For example, when 2 true breeding parents for 2 traits are crossbred, they produce 100% heterozygous offspring for both traits. When the F1 generation self-pollinates, there are 4 combinations of alleles per parental gamete, and the resulting Punnett square of these crosses would have 16 boxes instead of 4, as with the monohybrid crosses. Watch the linked video for more on dihybrid crosses. Years after Mendel studied pea plants, other scientists used his work as a foundation, realizing more complexity existed in inheritance patterns of traits. Mendel only realized through his work the idea of dominant and recessive traits. However, in the research since Mendel's time, the incomplete and codominant inheritance patterns have been discovered. Incomplete dominance is where 2 dominant traits blend characteristics. One example is crossbreeding a dominant red flower with a dominant white flower and producing offspring with pink flowers. This is a case where parental trait expression is shared or blended. With codominance, both dominant traits are expressed. In the case of the dominant red-haired cow crossbred with the dominant white-haired cow, both alleles are expressed as a mixture and the offspring has roan-colored hair. The written expression of these traits includes both capital letters to represent the dominance of both alleles, but then they carry a superscript to distinguish the color from the other dominant alleles. In diploid organisms that have 2 copies of chromosomes, one from each parent, it is only possible to inherit 2 alleles. However, more than 2 possibilities may exist in a population. Consider blood types. There are 3 allele types. There's the IA, IB, and recessive I. Depending on the alleles passed down from parents, offspring can inherit the A, the B, a mix of A and B, or if both recessive alleles are inherited, they end up with blood type O. Blood type is an example of codominance, where if both blood type A and B are inherited, both are expressed. In many species, female offspring inherit 2 X chromosomes, one from each parent, while males inherit an X from the mom and a Y from the dad. These chromosomes are not homologous as they are different lengths, and the X chromosome carries more genes. As a result, when genes are sex-linked, they are usually on the X chromosome from the mom and are therefore known as X-linked. Since girls inherit 2 X chromosomes, many times they are carriers of a gene and it can be masked by the allele from the other parent, while boys tend to be affected by the gene more often since they only inherit 1 copy of the X chromosome. When only one allele is inherited in this manner, it is known as hemizygous, hemi meaning half. With X-linked traits, it depends on whether the male or the female carries the trait and whether the trait is dominant or recessive on how it will be passed down from parent to offspring. If the female is homozygous dominant, then all offspring will inherit and express the trait. However, if the male carries the dominant allele on the X chromosome, then only his female offspring would inherit and express the dominant trait, but his male offspring would not inherit the trait at all. In heterozygous females, her offspring would inherit one of her X chromosomes and could inherit either the dominant or the recessive allele, producing a 50-50 chance she could pass down a sex-linked trait. In this example of a mother who is a carrier of a recessive gene, she could pass her X to a daughter or a son. If she passes the allele to a son, he will express the trait because he has no other X chromosome. If the father is the recessive carrier and the mother is not affected by the trait, then all the daughters will inherit his X chromosome and be carriers, while none of his sons will be affected since they only will inherit his Y chromosome. Watch the video for more on sex-linked traits. When 2 genes are closely located on a chromosome, they have a tendency to be inherited together. Notice the chromosome labeled linked on the left that has 2 genes with loci that are very close and therefore will tend to end up in the same gamete even if crossing over of genetic material occurs. The chromosome on the right, however, would represent an example of 2 genes not that closely located and therefore would not be linked and could be inherited independently of each other. In addition to the ideas of dominant and recessive, or incomplete dominance and codominance of alleles, there is another inheritance pattern that can influence the expression of traits. When one gene's allele masks the expression of another gene's allele, the ultimate expression of the 2 traits is influenced, and it's known as epistasis. In the case of the 3 dogs, there are 2 genes that influence coat color. One gene is for the color of the hair, while the other gene is for the expression of the hair color. When the black or brown gene for hair color is blocked by whether or not the gene or the coat color is expressed, the result is yellow coat color. The same idea occurs in the 3 colors expressed in mice. And we've made it to the end of chapter 8. So I wanted to remind you to go back and look at the review questions at the end of the chapter, and also to remind you that your second test coming up in about a week will cover chapters 6 through 9. So we have one more chapter after this one before your next test. And then to remind you too that you're over halfway through the summer session, so hang in there, keep turning in your discussion posts and your photos and your labs. And if you have any questions, let's just make sure that you let me know. All right.